everyone, or whenever you're watching this. Welcome back to the Pathogems podcast. I am your co-host number one, Caroline Clark.
And I am your favorite co-host, Erin Wright. And since today and the rest of this month of March is National Women's History Month, today we're going to be interviewing some very special women in science.
And specifically, we are going to highlight three different women involved in science, and they're all in different stages of their career. So today we will have one grad student, one postdoc, and then one PI.
Yes. So our first guest today is Dr. Chris Angel Lopez, who is a postdoc in the Geest Lab studying mosquito-borne viruses and how they move through wildlife and ecosystems. Her work focuses on arboviruses, including Cache Valley virus and La Crosse virus. Through field and lab work, she helps scientists better understand how these pathogens spread and how emerging diseases can be predicted and controlled. So everybody give a big Pathogems welcome to Dr. Chris Angel Lopez. Okay, so welcome to Pathogems. Yay!
This is Dr. Chris. Do you go by Chris Angel or Chris?
I usually go by Chris.
Dr. Chris Lopez.
Dr. Chris Lopez.
Welcome to Pathogems. Thank you for coming.
And so our first question that we would like to ask you today is kind of just like what drew you to virology?
That's a multifaceted question. So I'm originally from the Dominican Republic, and I moved to the U.S. when I was 7. Mm-hmm. Prior to that, there was this huge hurricane in the late 90s. And there was a dengue outbreak, and I was really little. All I remember was being in the hospital for 2 weeks. I didn't really know what was going on. I was just really sick. Turns out I had dengue. I mean, growing up in the Caribbean, you get a plethora of pathogens. So I was hospitalized multiple times for different things. Dengue, amoebas, a bunch of different pathogens. So that's quite normal, actually. So then I moved to the U.S. And I realized I was kind of good at biology, and that I really liked epidemiology. I went to a lecture my senior year, my last semester of my undergrad. I was like, I'm going to med school, like, I'm going to be a pediatrician. My best friend's a pediatrician now. We're like, we have a path, and we're going to stick to it, and we're going to do it together. And then I went to a lecture by this professor at Columbia called Ian Lipkin, and he's a microbiologist. And did you guys ever watch Contagion?
No, what is that about?
It's a movie about essentially epidemics and pandemics and stuff. But he was the lead advisor for that.
Okay.
And he won an award at my university, and we were required to go to this lecture for a microbiology class, and it like changed everything for me. It was this one lecture, and I was like, I want to be a pathogen hunter. That's what I want to do. And then that just kind of opened up the path to, I want to work with arboviruses, I want to do infectious disease work. After my undergrad, I took a gap year and I moved to South America, and I studied pediatric infectious diseases. And then I got curious again, and I was like, this is great, but now I want to see what happens on the side of the wildlife, because I'm like, I want to learn all the different components. And so then I went and I moved to Oklahoma, and I started doing my master's in biology. But it was mostly focused on wildlife infectious disease transmission. During the time, I met my PI, who then, he and I became really good friends, and then he started his own lab, and then I kind of joined his lab. So that's how I ended up here at BT.
Fantastic.
So you did your graduate degree, your first one at Oklahoma.
University of
Oklahoma, and then you did your PhD here at BT, and you stayed in the same lab for your postdoc. Can you talk a little bit about like that process of like transitioning from the graduate student to the postdoc, like in the same lab, and kind of like?
So funny. I still feel like a grad student. The responsibilities are different. But like, I guess the work is very similar. It hasn't really changed much, other than like I'm now like the lead in a bunch of different
Okay.
projects.
Yeah.
And I get to mentor grad students and undergraduate
Avery's
students.
behind the camera, guys.
Avery.
Out of there.
But I get to mentor some of the grad students, which I kind of did as a senior grad student. But now like the role's a little bit different. I loved working with Jonathan. That was one of the reasons I came here. I had options to go elsewhere. But like he and I got along really well. One of the things I like about working in his lab is that I was never really treated as a student. It was more like we were partners. And so it was like I came in, I have ideas, these are the kinds of projects I want to do for my grad school, and he was like, how can I help you get there?
That's amazing.
And so it's a very different dynamic than what I had before. And so coming into it and like being treated like an equal from the beginning kind of made me want to work harder.
Definitely.
For sure.
Because I was like, oh my God, like I am responsible for these things. And like, I'm not just like a subordinate. Like, he told me from the beginning, essentially, like, at the end of the day, when you graduate, you're my colleague. So 20 years from now, when you have your own lab, like, you and I are collaborators, and why would I treat you any different? And so that kind of really set our relationship. And it really worked out because then I didn't want to leave. And then I was like, all these projects I've nurtured from like these little specks where like nobody knew what this virus was, and now we're funded for them. So like,
that's
just,
super
stay
awesome.
and keep doing the things.
That's amazing.
Did your research change any like when you did the transition from grad student to postdoc? Or are you like still working on the same projects?
The projects are different.
Okay.
Yeah, it's still I still work adjacently with the same virus. I definitely work with the same group of viruses. So we primarily are now working with bunyaviruses, which are the largest group of arboviruses, but also the least studied.
Awesome.
So, yay. And so primarily we're working with Cache Valley virus, which was my little baby. And then I'm lead a lot now on lacrosse virus, which is the leading cause of pediatric encephalitis through an arbovirus.
When you were working in South America with like the pediatric infectious disease role that you had, were you working with some of the viruses that you study now?
No, it was very different. So the project that I was working on essentially was looking at the effects of weather conditions on infectious disease outbreaks.
That's so fun.
So in the late 90s, there was a big El Nino event in Peru, and there was mass flooding, and they had lots of outbreaks of different pathogens, and there was also like arsenic poisoning. That was
great. I love that.
and a lot of that had to do with like the runoff. So the way that Peru is laid out, you have like the coast, you have the mountains, and then you have the jungle. Up in the mountains, there's a lot of coal and mining and stuff, and so that runoff caused arsenic to get into the rice paddy fields out on the coast, and so there was a lot of arsenic. And like bacterial pathogens because of all the sewage water getting mixed up with all the food and drinking water and stuff. And so we were expected to have another El Nino, La Nina event the years that I was there in Peru. And so we were doing like a pre, during, and post study looking at gestational growth, but also like post-birth, whether these kids had any developmental issues and things like that. So that was primarily what I was looking at. We were also going out to sewers and testing for antibiotic resistance, and also testing the rice paddy fields for arsenic.
That's awesome.
Yeah.
It's really interesting.
So not the same. We had some norovirus outbreaks, but I don't really work with norovirus. But it was primarily more of a surveillance-based study. But it was very rewarding. I got to hold people's hands while they were birthing their babies.
Oh my gosh. Yeah. That's super cool. What are your career goals after you complete your postdoc? Are you wanting to stay in academia and have your own lab? I think you mentioned possibly that, or something completely different.
I don't know yet. I think a lot of that's gonna depend on the job market.
Yeah,
I know that's right.
We have a lot of PhDs and not a lot of jobs.
Uh-huh.
I don't think I want to go into industry. I think that if I had to choose, I would probably go into government or stay in academia.
Okay, yeah.
I like the idea of teaching and having like students. I think it's very rewarding. But I also really like, this is gonna sound kind of weird, but like having a fixed budget, like
Yeah.
Yes.
not, you know, like in government, like they give you like a budget and you work with it and it's great. And you can still kind of apply for grants. So that kind of like that. It's, I don't know, I don't want to say it's less competitive, but it's less cutthroat.
Yeah.
Definitely.
And so I kind of like that, but I would also really like being a professor if I got the opportunity.
Yeah.
Great. And for our final question, what advice would you give to young women interested in scientific careers?
Well, I think follow your passion. You know, I knew I wanted to be in like the medical field from very early on. I just didn't know what. And like if, for instance, like what changed my mind was going to a lecture.
Yeah.
So like if don't be like dead set on what you want to do. Like if you see something that you're passionate about and you're like, wow, I really want to explore this because I think this is going to be the most fulfilling way to like live my life, then I say, you know, don't feel like you have to stick to a mold. And like go out and try things.
For sure.
Definitely.
Yeah, that's great advice. Yeah. Yeah.
Well, well, thank you so much for coming on our podcast. We really appreciate it.
Yeah, of
Yeah,
course,
thanks.
no problem.
Yay! Okay, our next guest is Shannon Carney, who's a PhD student in Dr. Kylene Kenhall's lab, and her research focuses primarily on mosquito-borne viruses that affect the brain and nervous system. Her research examines encephalitic alpha viruses like Eastern equine encephalitis virus and how they cause neurological disease. She uses animal models, such as mice, to study these viruses and how they invade the brain and trigger severe inflammation. So everybody give a big pathogens welcome to Shannon!
Yay, Shannon! This is perfect. Hey!
So here we have Shannon.
Thank you, Shannon, for
Thank
coming.
you, Shannon. Welcome to Pathogems. So the first question that we're asking everyone today is, what inspired you to pursue virology, and I guess specifically a PhD in virology?
Sure. So actually, for a long time, I wanted to be a veterinarian. My undergrad is in animal science, but while I was in undergrad, even though I still thought that I wanted to go to vet school, I worked at a research lab. We did antibiotic resistance research on dairies, so it was still within the animal science department. And I really liked that, and I liked that a lot more than I thought that I would. And so after that, I kind of switched gears and I went and did my master's in public health here at Virginia Tech with an emphasis in infectious diseases. And while I was doing that, I sort of became a lot more interested in the One Health aspect and sort of the interaction of like humans, animals, and the environment. After that, I went and worked on a dude ranch for a little bit, and then I circled back and I was a research manager because I knew that I was more interested in research, but I wasn't convinced I wanted to go to grad school yet, and so I worked as a research manager. We looked at novel chemotherapeutics to treat cryptosporidium in the calf model, so I was still staying sort of large animal. And while I was doing that, I was like, I think that I really would like to be able to feel like I have some sort of creative control and be able to develop my own science. And I am really, truly interested in this. And so that is sort of what brought me to applying to grad school.
That's awesome.
Where did you do your undergraduate at?
Here.
Oh, amazing.
You've been
So
here,
triple tech.
been here many a year.
She's committed. She's committed.
Big Okie girl.
Yeah.
What kind of caused you to switch from the large animal side to like the more human directed side?
I think I'm really interested in sort of the interface of human and animal health. I think especially on the veterinary side, that gets a little bit missed, where there's a lot of focus on the animal health, which is of course very important, but a lot of the research that's done for animal health has applicability over to the human side. And so I'm sort of interested in that interface, and also, as you all know, many diseases are zoonotic, and so it's really important for us to be able to sort of have that crosstalk between the two sides.
Yeah, that's
awesome. What is your current research project about?
So I'm in Kylene Ken Hall's lab, and I personally work with Eastern equine encephalitis virus. It's fairly rare, but highly lethal, so it's a 30 to 70% mortality rate. And of those who survive, 50 to 90% will suffer from long-term neurological sequelae, which can include schizophrenia-like symptoms, movement disorders, seizure-like behavior, and it's not really well understood why that happens. And so what we are looking to do is develop and characterize a mouse model of infection, both acutely and a sublethal infection, to be able to study both acute and long-term effects of infection to better understand and hopefully someday be able to insert vaccines and therapeutics into that model.
Yeah. Do you still get to work with the animal side of everything, like outside of the mice?
No, I only work with mice at this point because Eastern is what's called a select agent, and so the Department of Health and Human Services has determined it to be a risk for bioterrorism, and so it's very, very controlled environment in which we work.
Yeah,
Makes sense.
Makes sense. It does make sense.
Don't want to get involved with bioterrorism.
Not really.
Not really.
Yeah. So as a grad student, what does a typical day for you look like here?
There is no such thing as a typical day, honestly. Every day is different, every week is different, which can be a good thing or a bad thing depending on how you look at it. It really depends, like if I have a mouse study going on, I'm in the ABSL3 doing a lot of mouse work, and that takes up or prepping things for that, and that takes up the bulk of my time. But if I don't have a mouse study, then I'm on the back end, I'm working on processing those samples and getting that data together. So some days I'm doing a lot of mouse work, some days I'm just prepping tubes and getting things ready. Some days I'm finally processing data and getting to see what I've done. And some days it's like reading papers, and some days it's just like having collaborator meetings and figuring out the next steps for what we want to do. So every day is different, and I think that keeps things interesting.
For sure.
What year are you in your PhD?
I'm in my 4th year.
Nice. So, because you're a candidate now.
I am a candidate.
Oh, Queen. What has been the most challenging part of your PhD thus far?
I think the flexibility you have to have, the mental flexibility that you have to have with being willing to cope with the fact that things are not going to go the way that you hoped.
The majority of the time.
Right. No matter, and sometimes, oftentimes, it has nothing to do with you. And so you just have to deal with the fact that experiments are not going to go the way that you hoped or even thought they would, and you're going to have to figure out how to adjust to that. So I think it's really the mental flexibility.
I agree. Yeah, for sure. For sure. What kind of, I guess, like, taught you that, or do you have a story of something that was, like, frustrating and kind of how you got over that?
I think for me it's honestly because my path to grad school was fairly non-linear. I think that is what has taught me more mental flexibility and resilience than anything else. I didn't pursue my PhD right out of undergrad. I did go to grad school, but then I went off and did other things. And so I think that sort of offered me a lot more perspective. And so I wasn't just so focused on education and research and not failing and getting through things. And so I think that has offered me perspective that has been valuable to me in learning that.
Yeah. What advice would you give to students who are looking to start grad school or want to pursue a career in science?
I think I would tell them that they don't have to fit a certain stereotype to be a scientist. They also can go back to school even if they've been in the workforce, and that there is no one person that is a better scientist than anyone else. Everyone brings different perspectives.
I know that's right.
And so to not feel discouraged if they don't feel like they fit that mold, or if they're like, well, I'm like well past undergrad, I don't feel like I could go back. And so I want for people to feel like they can still do that and actually may even benefit from doing something like that.
For sure. Yeah.
Dang, that was good.
She booked that.
And then this is, it's very similar to the last question we asked, but as it is Women's History Month, when we're filming this, go March.
Yep.
What advice would you give to specifically young women interested in scientific careers?
I would say very similarly that you don't have to fit a certain mold to be a woman in science, and I think that it's the diversity of women in science that makes science great, and so I think that...
I need to write that down.
She's being so wise.
And so I think it's just important for people to remember that I think sometimes it's tough because you're like, well, like, so somebody that I know is working harder than me this week, like I'm not doing enough, and so women in science, I think it's good for us to remember that we're all working towards the same goal together, and that we should encourage each other and be collaborative and raise each other up while we're doing our science.
Great words.
That was beautiful.
I'm writing that down, and I'm putting it on a poster.
This is why we invited you.
I know that's right.
Yeah.
Amazing.
Okay. Well, thanks, John, for coming.
Thank you very much. Woo! Our third and final guest is Dr. Nisha Duggal, who is an associate professor of biomedical sciences and pathobiology at Virginia Tech, who studies emerging viral diseases. Her lab focuses on viruses such as West Nile, Zika, and Usutu virus, and how they evolve and spread between mosquitoes, birds, and humans. Through molecular virology and animal models, her team works to understand viral transmission and improve strategies for preventing future outbreaks. Everybody give a warm welcome to Dr. Nisha Duggal.
Yay! Thank you for coming on the podcast.
This is Dr. Nisha Duggal, everybody.
So, our first question for you today is, what inspired you to pursue a career in leading your own laboratory?
Yeah, that's a good question. So I made the decision, I thought about it for a long time. And then finally, I did my postdoc in the CDC, so in government, in Fort Collins, Colorado, and I loved working for the CDC. But after I'd been there for a few years, I made the decision that I missed students.
Yeah.
So there's not a lot of students at the CDC. There was a few, but I missed that. Surprisingly. So I decided to apply for faculty positions so I could lead my own research group and work with students. So that was really the decision point. But I was really, loved my experience as a graduate student and wanted to just really loved that sort of dynamic between mentor and student, and so wanted to pursue that.
Yeah. When you had students at the CDC, what kind of students were they? Were they like graduate students at, if it's in Fort Collins, CSU? Or?
Yes. So there was a student in the lab that I was in who was officially through CSU and did her PhD at the CDC. There was a couple of visiting scientists, visiting students that would come for a summer. And then, there were a few postdocs. I was a postdoc, and there is a program to do postdoctoral fellowships there. So there are more postdocs than there are students.
Yeah. And what, I guess, was that transition, from the postdoc to, having the official faculty role?
It was challenging.
Yeah.
That first year of starting up your lab is tough.
Yeah.
But I still remember the first time. So I don't know if you guys know Seth. He's a technician in my lab. He's been in my lab since the beginning. So I heard him when I was here for 3 months, and he's still in my lab.
That's awesome.
And we did our first plaque assay together.
I love
And
that.
I took a picture of it, and I still remember the excitement of being oh my gosh, we did it! We can actually grow virus and plaque it. So it was really exciting.
Yeah.
Challenging, but
For sure.
It was it was really exciting.
Yeah. Do you have that framed somewhere?
I have the picture. I actually included it in a lot of my talks, on the last slide.
Yeah.
I have a picture of the plaque assay, so that's the one. That's the one.
What is your lab currently studying in virology?
Yeah, so my lab studies RNA viruses, Mostly mosquito-borne viruses, but we also study respiratory viruses as well, thanks to COVID. And we're really interested in how viruses are transmitted between individuals and how they cause disease. So in terms of mosquito-borne viruses, we study like actual mosquito-borne transmission. We use mosquitoes and birds for some of our viruses, and I have one student that's working on sexual transmission of Zika virus. We use a mouse model for that, and then sort of the respiratory side, we work a lot with Lindsay Marr on campus looking at airborne transmission of viruses and pathogenesis of SARS-CoV-2 using a hamster model.
Yeah.
Hamster.
Yes.
Whoa.
Yeah. So
I didn't know that was a thing.
The birds didn't elicit a
yeah, usually it's the birds. Yeah.
Well, you know how I feel about a bird.
She's not a fan.
There are mixed opinions.
They scare me.
Yeah, very terrified of them.
Very, very.
Yeah. So with that research that's being done in your lab, kind of like what role do you play in it? Is it more administrative, or do you get to do any research now?
I do still do. I'm doing an experiment right now. Counting my plaques. I don't. It definitely has dropped off. When I first started the lab, I was working in the lab every day, and it has decreased so that my students are doing the experiments, and I do sort of exploratory experiments that are like, maybe just like I have an idea and I really want to test it right now, or something that I'm not sure is going to work.
Yeah,
I'll try it. Mostly, I spend my time writing grants, teaching. Advising my students. Which I love all of those things.
Yeah.
I do help out with animal work. That's kind of one thing that I still do a lot of. So with the bird experiments, I love the bird experiments. So whoever's the student working on the bird experiments, they get to work with me a lot. So, yeah.
When you do the bird experience, like what does that entail?
Yeah. So these are experiments done at BSL-2. We have like isolator cages. Right now we work with songbirds.
Okay.
We have worked with chickens in the past. Baby juvenile chickens.
They still scare me.
Okay.
I don't care what size they are.
Pretty cute. So right now we work mostly with canaries and house finches. So we, if they're house finches, they're trapped in the wild, and that's in collaboration with Dana Hawley on our campus. Canaries we can purchase, and then we inoculate them with a virus in the lab. We can monitor their health. We collect blood. That's the tricky part, is collecting the blood from them. So we're able to do, what I like about it is that songbirds are the natural reservoir for a lot of the viruses we study, and so being able to actually put the virus in the right host is just like,
that's amazing.
Yeah. So that's what I like. and I like the birds.
Yeah.
This is gonna be a really weird question. How much does one bird cost?
They can be expensive.
Okay.
Canaries are expensive, so I think if you were to go to like a store, it costs $150. Sometimes we can get them for cheaper, but
Dang.
they are pricey.
That's crazy. What kind of has been like your experience as a woman in science?
My experience has been really positive. I've had really supportive mentors since I started working in a lab as an undergrad, as probably you did too. Worked in several labs as an undergrad. I worked as a technician for a couple years, and then grad school and postdoc. So I've loved it. I've learned a lot. My mentors have been fantastic.
That's
so
amazing.
It's been a really positive experience.
That's great to hear.
Great.
Yeah.
What advice would you give to any young women interested in science careers?
I would say go for it. My experience has been very positive. I think, so my experience has been very positive, and I would expect that another young woman going into science would have a positive experience. So, there can be ups and downs in science, but to not be dissuaded by setbacks, let's say, I don't know, you apply to grad school, get in the first time. That doesn't mean that you shouldn't go.
Yeah.
Like it took me 2 rounds to get a faculty position, 2 years of applying for positions. So, I think that's a normal part of the scientific process. It's also part of the career process. So, just to, if something is important to you, to go for it.
Yeah, for sure. That's great advice.
Yes, definitely.
Yeah. And so our last question is one that's been really on our minds personally, and we've been having a debate about it. But if all of the flying birds were to go missing in the world, and in this hypothetical universe, no one is communicating
No
to you
one's
about
talking
this.
about it. There's no,
No one's
like,
talking about it.
There's no
They're
like
gone.
environmental shit. Like the
Nothing.
food chain is fine.
It's fine. But like the ducks, geese, they're all still there. I guess
Okay.
they can maybe fly, but they like the ground more.
Yeah.
How long would it take you to notice?
How long did it take me to I think I would notice pretty quickly.
Okay.
I agree with you.
Yeah.
See, I'm terrified of them. Her original answer was that it was going to take her a year.
I think it
But
would
now
change
she's
it
went
now.
down to a month.
I think it would be a month, which is
Okay.
still a long time, and it's kind of embarrassing to admit, but I don't think I'd notice initially.
I think that the sounds.
That's
Yeah.
the sounds
I
and the
think
fact that
even-
I'm not scared when I walk outside.
Yeah.
You know?
Yeah.
So we've got,
Yeah.
within a day, you're thinking an hour.
Within
I'm
an
pointing
hour.
at Avery.
I thought you were gonna ask me like, what do I think killed the birds?
No, but what do you think killed the birds?
Yeah.
I mean, West Nile could be part of it, but that's
There
what
we
they
go. You heard
back
it
to
here
research.
first.
just
Back to the
just
research of it all.
crows.
Yeah.
Just the crows. Yeah.
Okay. Pathogens out.